こんにちは、先生。
サワディクラブ、こんにちは。
久しぶり。
久しぶりですね。元気ですか？サバイディールか？
はい。先生、今日もありがとうございます。
あ、いえいえ、はい。
今、研究室ですか？
でも研究室ですね。はい。
今、夏休みですね。
まあ、そうですね。今は授業が日本はないですね。9月の終わり
え
ぐらいからですね、また。
ーえ
はい。
ーえありがとうございます。
で、
このセミナー、ありがとうございます。
これ授業ですよね。授業ですかね。なんかセミナー。
えーせあのせ授業のセミナーです。
あ、授業の方ですね。はい。
えー
わかりました。
ま
大体まあ、1時間か90分ぐらいですかね。
あ先
はいはい。
生次第の、not serious。
わかりました。
こうゆっくり。あと、多分、
うん。
学生さんが多分十何人くらい
ありがとう
来るかと
ございます。
思いますけ
ちょっと
ど。
私の研究が難しいかもしれませんけど、なるだけ
え
頑張ってありますね。はい。
ありがとうございます。少
はい。
しでも。
はい。OK です。
はい。
はい。
まあまだ時間がありますけど。
うん。ですね。はいはい。ちょっとまだ待ちますね。
สุมนมานบิ๊กบอ
Hello,
สบิ๊กบอส
good
はい。
afternoon สวัสดี
สวัสดี
ค่ะ
สวัสดีครับสบายดีไหมครับ
สบายดีค่ะ very good thank you for give a special lecture for MUTT again あ、そう先
はい。
生
はい。Yeah. Yeah,
I'll
thank,
send maybe
yeah, thank you again for giving me this great occasion. Yeah, to talk my research work. Yeah, thank you so much.
先生まだ時間がありますので、多分学
あ、
生さんが多分
はい。
13時35分か40分くらい来るか
あ、大丈夫。
もしれない。
はい、大丈夫です。はい。いや、ゆっく
あ、
り来てます。はい、
ありが
OK
とうご
です。
ざいます。
ありがとうございます。
なんえ、台風もう
うん。
少し来ます台風
あ、そうですね。日本は台風が今多いですね。うん、今ね、ちょうど日本に大きなビッグ台風が来てます。
うわ僕ちょうど明日、日本、
も
札幌の方に行きます。
う、あ、本当？
北海道情報大学かな。
あ、はいはい。何先生ですか？芦田先生、花田先生。
花田先生です。
花田先
あの
生ね。あ、
昨日、一昨日かな。一昨日じゃなくて、金曜日かな。日、花田先生、このセミナーも発表しましたので。
あ、そうですか。おー、すごいですね。
今日は、ビッグボス、そう。
いやいやいや。そうですか。いや、すごいですね。北海道ね、多分大丈夫だと思いますよ、まだ。来週、いつく、明日来るんですか？明日日本ですか？
明日、夜便に
おー、
来
うん。
ますんで、ちょうど
来る。
台風が、
あ、そうですね。ちょっとね、もう気をつけて来てください。
はい、まあ、キャプタン次第。
うん。
キャプタン次第。はい、わかりました。
はい。
先生、
また。
先生のスライドを
うん。
シェアできます。シェアス
はい。
ライ
はいはいはい。シェアしましょうね。はい
ド。
は
はい。
い。えーっと、
OK。
はい。
QR コード入って。
はいはい。おー、ブンヤン。
Hello.
はい。サバイリーマイカ。
Nice to meet you online. Nice to meet you online, sir.
Thank
Thank you
you.
for coming. Thank you. Thank you.
Yeah,
So I
thank
hope to come you in Thailand soon.
If I have a
Okay.
chance.
Sorapong Sang, he is traveling.
Yeah, yeah,
He
yeah.
is he is my boss now.
Yeah, yeah, yeah.
He is a very big man and busy. So thank you. How are you, sir? How are you doing? Also.
Yeah, fine. I'm really good.
Good. Okay, I may leave you. I just come to say hello to you.
Okay. Thank you so much.
All
Yeah.
participants are member and also PhD student.
Okay.
Very interesting topic from you.
Okay.
Thank you.
Yeah,
Okay.
thank you so much. Yeah.
Also, Sensei, one more information.
Hi.
Dr. Suman now, she is very beautiful.
Yeah, I know.
Of course,
Is very
it's fact.
beautiful
It's
now.
fact. It's fact.
She is not fat anymore.
I know well.
Okay, Sensei. Now student maybe at
Hi.
totally about 10.
Hmm.
They can come or not, I don't know. So today we would like to say thank you very much for Sensei for this seminar. ありがとうございます。どうもおおきにとか ก็นักศึกษาครับก็วันนี้ก็เป็นอีกหนึ่งโอกาสที่ดีนะครับโปรเฟสเซอร์อาโศกนะครับแห่ง KIT Kyoto Institute of Technology นะครับก็อยู่ในเมืองเกียวโตนะครับก็เซนเซเคยมาอยู่กับเราประมาณสัก 3-4 เดือนนะครับแล้วตอนนี้เซนเซกลับไปเป็นโปรเฟสเซอร์นะครับที่ญี่ปุ่นครับแล้วก็จะก็เป็นโปรเฟสเซอร์ที่ใจดีแล้วก็รับนักศึกษาเราเข้าไปทำแลบด้วยแล้วก็มีอาจารย์เราจบจากแลบท่านด้วยนะครับเพราะฉะนั้นก็เป็นโอกาสที่ดีนะครับที่จะได้ฟังงานของอาโศกเซนเซนะครับโอเค so today thank you very much for your presentation at the seminar course of energy and materials engineering so please let's say not serious because this is for master course they just come MEG M1 master course
Okay. Yeah,
so
thank
it's
you
your
so
turn
Okay. Thank you,
Thank you very
Professor.
much.
Yeah, Ajahn Surapong. Yeah. And first of all, so I'd like to express my great thanks to my friend, my best friend, yeah, Professor Surapong, yeah, for giving me this great occasion. Hello, everyone. So I'm Yuji Aso, working at Department of Biobased Material Science, Kyoto Institute of Technology, Japan. Then today, so I'd like to talk about my current research work on the microbial production of monomers for the synthesis of bio-based polymers. Because so my major is the microbiology, yeah, and covering the polymer science as well. So I'd like to focus on how to produce the polymer feedstock, so-called monomers, by using the microbial production from biomass. Okay. Then before my talk, I'd like to introduce my university briefly. Kyoto Institute of Technology is one of the national university in Japan, locating on the Kyoto City, the former capital of Japan. Our university is dedicated to the work with education and research on the interdisciplinary field of art and science by 6 academic fields, yeah, like them. Of which I'm belonging to the Faculty of Fiber Science and Engineering. This university consists of around 4,000 students and 300 faculty members, meaning this university is very compact, relatively. Then, as you know, the Kyoto is a very famous city with sightseeing, so I welcome you all to visit Kyoto, yeah, for sightseeing, and our university as well for any collaboration as well. I welcome anytime. Then for education, I'm working with the Department of Biobased Material Science, which is dedicated to the interdisciplinary field, yeah, especially education and research by integration of the 3 fields, biology, chemistry, and material science. For research is on the biomass-derived product, so-called bio-based materials. Then our department consists of around 70 students and 12 faculty members, including me. Our department is recruiting now the graduate students and collaborators anytime, so please take a look at our web page if you are interested. Okay, so I'm entering to today's topic on the bio-based plastics. During several decades, for us, it is easy to imagine that so many plastic products surrounding us and supporting our life and human activities. Consequently, very massive plastic waste have been generated, resulting in serious environmental issues to be solved soon. In other words, I can say that the Earth is becoming a plastic planet. In terms of plastic pollution, I would like to touch with some data on that. Currently, global production of major plastics, mostly polyvinyls, reaches around 400 million tons a year. And the production is predicted to be up to twice in 2050. Currently, two major issues, global warming and marine plastic waste, have been recognized, especially total amount of microplastic, marine plastic waste is predicted to be the same as that of the fish in the ocean in 2050. Also, the plastic wastes are believed to cause microplastics problem, leading to harmful diseases for not only marine animals, but also human beings. To solve these problems, the world adopted zero plastic waste declaration toward achieving SDGs. As one of the solutions for these issues, we can suggest the use of the biomass plastics, which are derived from renewable resources. Biomass are predicted to be a sustainable and carbon neutral process. What we can see here is a life cycle of polylactic acid as an example. In this case, polymer feedstock, lactic acid can be obtained by microbial process, so-called fermentation, and then polymerized to polylactic acid after purification from microbial culture. Polylactic acid works as one of the biodegradable biomass plastics, especially under special conditions like high humidity and temperature. Biodegradability can be explained based on the type of polymer backbone. Major plastics consist of the polyvinyls, possesses the irreversible CC backbone, resulting in generation of plastic waste problem in the environment. On the other hand, polyesters such as polylactic acid possesses a reversible ester bond showing biodegradability. Therefore, such biodegradable function is ideal for reduction of plastic waste remaining in the environment. Briefly, plastics can be categorized into the four types according to their properties. Upside and downsides are plant-based and oil-based, and left side and right side are non-biodegradable and biodegradable plastics, respectively. From this categorization, we can say that more sustainable and eco-friendly plastics are plant-based biodegradable plastics, such as polylactic acid, PLA, polyhydroxyalkanoates, PHA, and polybutylene succinate, PBS. So I'd like to talk current trend in Japan toward achieving sustainability from the aspect of biomass plastics. In Japan, as well as other countries, it is becoming more easy to find some product made from bioplastics. There are two major associations, certificating the biomass plastics by these marks. More recently, Japan Bioplastics Association developed new marks for biodegradable plastics to keep up with the times. Also, the government of Japan made a plan to introduce 2 million tons of bio-based plastics to the market by 2020. Along with this, to reduce 2030. Along with this, to reduce the marine plastic waste to net zero by 2050, the government of Japan enacted Osaka Blue Ocean Vision with Marine Initiative, like that. Okay, let me move on to the topic on bioprocess using microbials. They are very tiny and invisible, but have the great potentials, such as rapid growth and easy gene manipulation. While their growth, substances such as biomass can be converted to the variable products like lactic acid. This can be shown as this diagram. So microbial process have some advantages such chemo, stereo, and regiospecificities during reaction under moderate conditions. This realized green chemistry by eco-friendly process and produces chemicals which can hardly be synthesized by chemical reactions. For example, lactic acid can be obtained from lactonitrile industrially, but the resulting product is a racemic mixture, which is not preferable as a polymer feedstock due to chemical reaction. So, for separation of D and L forms, we have to use a microbial process based on the enzymatic reaction. Here we can see the diagram of the strategy of microbial engineering for bioprocess. First, microbials can be modified genetically by introduction of genes involving in the production of target chemical and disruption of genes involving in the production of byproduct. This genetic manipulation is known as metabolic engineering. After this, the resulting microbials can be subjected to the fermentation under optimal conditions such as the nutrients, pH, and temperature in order to realize a higher production. This research is known as fermentation engineering. By combination of the metabolic engineering with fermentation engineering, we can produce target chemicals from biomass effectively using microbial process. In my laboratory, I'm doing two research topics related to monomer production from biomass using microbials. First one is on vinyl monomers, such as itaconic acid, and harboring a terminal double bond, CC double bond, like that. To date, I succeeded in isolation of the new microbes producing such chemicals from nature and production of itaconic acid from carbon dioxide using engineered photosynthetic microbes. Second topic is related to today's talk regarding diol production. Diols are polymerized with dicarboxylic acid, resulting in the polyesters. If diols can be obtained from biomass, the resulting polymers are eco-friendly. Today, I'd like to talk my current research works on two diols. 1,2-propanediol, and glyceric acid, how to produce them by microbial process. So first topics is on the 1,2-propanediol, 1,2-PDO. Then 1,2-PDO is a promising diol for many industrial applications. There are two forms, R and S, but to realize the high crystallinity of polymers consisting of 1,2-PDO, optically pure 1,2-PDO is required. So microbial process is suitable for the production. So interestingly, some microbials in nature can produce the 1,2-PDO, but the production titer is very low, meaning that appropriate genetic modification is required if use microbials. As mentioned, conventional chemical reaction generate optical impure 1,2-propanediol from oil via high temperature process. This means that this process is not ideal for eco-friendly and production of polymer feedstock. Here, I propose the biological production of optically pure 1,2-propanediol from biomass via green and simple process. Eventually, they can be used as a polymer feedstock because of separately producible. So in this study, I aimed to develop bioprocess of 1,2-propanediol production. For metabolic engineering, I designed for metabolic pathway like here. Glucose was used as a first carbon source, and then lactate. This is the intermediate metabolite, works as a precursor metabolite of 1,2-propanediol. As production host, so Escherichia coli and Lactococcus lactis were selected as a D-lactate and L-lactate producers, respectively. Escherichia coli, E. coli, possesses an endogenous pathway to reduce the lactate production. Lactate production by PFLA gene. So I disrupted this gene to avoid the consumption of the produced lactate. To add the external pathway for 1,2-propanediol generation from lactate, three heterologous genes, PCT, PDUP, and YAHK were introduced into these hosts. First of all, I investigated the function of the 1,2-propanediol pathway in E. coli. As expected, the recombinant E. coli could produce the 1,2-propanediol at 1.3 grams per liter after 96 hours cultivation, along with the intermediate lactate production. This showed that the introduced pathway working with the 1,2-propanediol production from glucose worked well. Next, to enhance the production, I performed to produce the 1,2-propanediol using lactic acid bacteria, since they could produce the high amount of lactate from glucose. Especially Lactococcus lactis produces L-lactate without byproduct and can be easily genetically modified. To produce the D-lactate in Lactococcus lactis, L-lactate dehydrogenase gene on the its genome was replaced with the D-lactate dehydrogenase gene from the D-lactate producer, Lactobacillus delbrueckii, by homologous recombination, resulting in the construction of the new D-lactate producer named AH1. Ideally, the resulting strain produces the D-lactate instead of the L-lactate from glucose with a high purity. Using the resulting recombinant Lactococcus lactis strains, 1,2-propanediol production was evaluated. As a result, the strain producing the L-lactate generated S-form 1,2-PDO with approximately 95% purity, optical purity. Vice versa, the strain producing the D-lactate generated R-form 1,2-propanediol with 78% purity. From this observation, we can say that introduction of the 1,2-propanediol pathway into lactic acid bacteria enabled us to produce 1,2-propanediol from biomass separately. After demonstration of the metabolic engineering, I performed fermentation engineering based on redox balance. So redox means reduction and oxidation. For enzymatic reactions go smoothly, enzyme cofactors such as NADH and NADPH must be maintained balance during metabolisms. But 1,2-propanediol pathway requires these cofactors to activate itself. To balance the cofactors, I demonstrated to use the other carbon sources, gluconate and mannitol, which can be generated further NADPH and NADH respectively compared to glucose. To prove the hypothesis, the recombinant Lactococcus lactis was incubated with a series of carbon sources. As expected, addition of both gluconate and mannitol improved the 1,2-propanediol production, suggesting these carbon sources balanced the redox balance, and consequently the production was improved. Unexpectedly, only adding the mannitol resulting in the highest production of lactic acid, but not 1,2-propanediol. The reason why this strange result was obtained is excess NADH generation, by which lactate from pyruvate is promoted. What we can see is the production curve of 1,2-propanediol when the recombinant Lactococcus lactis was used. This strain produced 1.2 grams per liter of 1,2-propanediol after 72 hours cultivation. The production titer was around twice higher than the when glucose was used. Lastly, pH control fermentation was demonstrated because the pH of culture dropped due to the production of lactic acid and consequently cell growth, and 1,2-propanediol production ceased. To maintain the culture pH, acid solution was added to the culture during the cultivation automatically. As a result, 98 hours cultivation resulted in the 1.9 grams per liter of the 1,2-propanediol using this kind of the jar fermenter system. This is the first demonstration of the 1,2-propanediol production using lactic acid bacteria. Okay, let me move on to the next topic on the glyceric acid. So glyceric acid are categorized to the diol group, but it possesses a carboxylic acid group, carboxylic group. There are two forms, S form and R form, but biological process generate R form only. It is known that acetic acid bacteria are able to generate glyceric acid from glycerol with high purity, which is unknown mechanism. So genetically modification is not applicable to acetic acid bacteria for now. Glyceric acid is a promising chemicals as a polymer feedstock, like here, but that is not used as the industrial application yet. This may be due to not well-known chemical. In addition, enzyme alditol oxidase can be directly convert glycerol to glyceric acid by oxidization. As conventional method, glyceric acid can be obtained from glycerol, via bioconversion process using acetic acid bacteria. However, glycerol is uncommon carbon source, and this process requires relatively high concentration of glycerol. On the contrary, I proposed new method for glyceric acid production from glucose through glycerol. The intermediate using recombinant E. coli, Escherichia coli with a high potential for fermentation. By introduction of the two genes, GPD1 and GPP2 into E. coli, glycerol was generated from glucose. In addition to this, glyceric acid was generated from glycerol by further introduction of LDO. LDO encodes the alditol oxidase from other microbe. So for metabolic engineering, I designed for metabolic pathway like here. Glucose was used source. The glycerol work as the intermediate metabolite, for production of the glyceric acid in recombinant E. coli. Then this study, I selected the E. coli because the E. coli showed the very rapid cell growth and easy gene manipulation. Then E. coli transports the intracellular glycerol and consequently less production of glyceric acid. To avoid this, the transporter gene, GLPF, was disrupted. Along with this, endogenous pathway conferred by the GARK-GLXK gene to assimilate the resulting glyceric acid was also disrupted. To add external pathway for glyceric acid generation from glycerol, a heterologous gene LDO encoding the alditol oxidase was introduced into the host strain. So first, gene introduction was evaluated. It was confirmed that introduction of the GPD1 and GPP2 resulted in the generation of the glycerol from glucose. And the introduction of the LDO resulted in the generation of the glyceric acid from glycerol. This result suggests that the introduced genes functioned in E. coli as expected. Next, gene disruption was evaluated. When a set of 3 genes were introduced simultaneously, glyceric acid was generated from glucose directly. To improve the production, I further disrupted the related genes, reducing the glyceric acid titer. Unexpectedly, disruption of GARK and GLXK genes led to the less cell growth due to the non-energy generation. On the other hand, disruption of the GLPF along with the GARK and GLXK led to the less cell growth, but a bit higher production of glyceric acid. This result showed that the production titer was decreased by the gene disruption, but specific productivity was improved. I demonstrated the fermentation engineering following to the metabolic engineering. Gene disruption resulted in less cell growth, as mentioned. To improve the cell growth, amino acids were added to the culture, resulting in the recovery of cell growth. Then, during the fermentation, hydrogen peroxide was generated because LDO generates the hydrogen peroxide. Then to eliminate this resulting hydrogen peroxide, so I added the manganese to the culture since the hydrogen peroxide showed the toxicity against Escherichia coli. As a result, cell growth and glyceric acid production were well improved. Lastly, pH control fermentation was demonstrated because the pH of the culture dropped due to the production of glyceric acid and consequently cell growth, and glyceric acid production were inhibited. To maintain the pH of culture, alkali solution was added to the culture during the cultivation automatically using this kind of the jar fermentation system. In addition, a mineral-rich medium was used for further support for cell growth. As a result, only 36 hours of cultivation resulted in the 2.4 grams per liter of the glyceric acid generation. This is the first demonstration of the glyceric acid production from glucose directly. Prior to use the product produced in the microbial process, the purification step is mandatory for separation of the target chemicals from the other impure compound in the culture. This process is so-called downstream process. And typically, multiple steps are combined in order to obtain the maximum yield and purity of the product. Academically, unique technique is applicable to the separation of the process. In the case of separating the diol, boronic acid, it is known that boronic acid interacts with the diol under alkali condition selectively, resulting in the boronate ester. This reaction is reversible, meaning diol are released from the complex under the acidic condition. Using boronic acid with hydrophobic moiety, reactive extraction of diol with organic solvent can be done. In this study, I demonstrated the separation process for glyceric acid. What we can see is an illustration showing the model of the reactive extraction separating glyceric acid using naphthalene boronic acid, NB, and trioctylmethyl ammonium chloride, TOMSC. Glyceric acid in the culture after fermentation process can be extracted by the interaction with the NB under alkali conditions, leading to isolation into the organic phase. This step is reactive extraction. After this, when newly adding acidic solution, glyceric acid can be released from the organic phase to the aqueous phase, resulting in the separation of the glyceric acid. Genetically, TOMSC works as a phase transfer catalyst helping out diols with the moving to the organic phase. Okay. To prove this mechanism, first I demonstrated the separation of the glyceric acid from aqueous solution without any contaminants by changing the concentration of naphthalene boronic acid. Hexane and octanol were used as an organic solvent. As a result, when no naphthalene boronic acid presence, glyceric acid was separated to some extent due to non-selective extraction. On the other hand, by adding the naphthalene boronic acid, improved the yield in dose-dependent manner. No adding TOMSC resulted in no separation. These data show that glyceric acid was extracted by naphthalene boronic acid and TOMSC in a coordinated manner. Next, I evaluated the separation of the glyceric acid from model microbial cultures in the presence of the some carboxylic acid, generally generated during fermentation. Interestingly, other carboxylic acid has an insignificant effect on the glyceric acid yield at nearly 60%. Regardless of the type of the carboxylic acid. However, all the carboxylic acid used in this study were extracted due to the non-specific extraction. Especially, tartaric acid is not affected by other carboxylic acid, but some of them is extracted simultaneously. So, it was revealed that an appropriate pretreatment for elimination of these impurities is required before this separation process. Lastly, separation of glyceric acid from microbial culture was demonstrated. The culture prepared from glycerol using acetic acid bacterium was used to eliminate impurities such as glycerol as the substrate. Ethanol precipitation was performed. HPLC analysis showed the culture before separation contained acetic acid at non-negligible level. On the other hand, separation resulted in the improvement of the purity in a certain degree. Although there is still room to reduce loss of the glyceric acid. For continuous process, recycling the organic phase was demonstrated twice. Interestingly, the recycling demonstration showed non-significant change in the yield. From this result, this process is cost-effective and applicable to continuous separation process. I summarize here the obtained result. By combination of the metabolic engineering with fermentation engineering, 1,2-propanediol and glyceric acid were produced from glucose directly using microbial process and can be used as a polymer feedstock, bio-based especially. Then hopefully these techniques developed during these studies would pave the way to produce the other diol compounds from biomass via eco-friendly and sustainable process, and also would help spread biomass-based plastics having the biodegradable function in the environment. So I'd like to close my talk after expressing the special thanks to many collaborators. Thank you for your kind attention. So I would like to close my talk. Then if you have any question and comment, would you give me your comment or questions?
Okay, Doctor, thank you very much for your presentation. It was
Hey,
very nice.
Sensei, thank you very much. So
Ah, not that.
now it's time for the question. Sorry.
Okay, no problem.
Okay, Nima, he's my PhD student.
Yes, okay, please.
Okay.
Yeah, thank you very much for your presentation. I have it's a like general question, like how does the microbial production of bio-based monomer like glyceric acid compare in terms of cost, stability? If you want to elaborate it for factories, how can this change the environmental impacts and traditional chemical synthesis methods? How can we compare these?
Yeah, thank you for your great question and comment. Yeah, as you know, the microbial process is not essential, but that I believe that this is the alternative technique to produce the polymer feedstocks. And then typically the microbial process is believed to be a more eco-friendly process, because the microbials require the moderate temperature and aqueous conditions, yeah, compared to the chemical reaction. Like that, you know, the chemical reaction requires a high temperature, high pressure, and extreme condition like strong acid condition or something like that. But these conditions show the environmental impact, right?
Yes.
Then, yeah, they compared to that reaction, yeah, the microbial process show the more moderate condition, okay, with that organic solvent and low temperature, something like that. So I believe that, okay, the how can I say, the bio-based materials are eco-friendly, but if we produce that materials via the non-eco-friendly process, okay, the resulting materials are not eco-friendly, I believe. So we have to combine the eco-friendly techniques, okay? Not only materials, but also the process using like a microbial process, something like that. And then I illustrated the characteristics of the microbial process like that. Okay, so we can also realize how different the properties, yeah, of the microbial process and the chemical process, like that.
Oh, I see, I see. So somehow we need a combination of these two in a process.
Yeah. And then also the microbial process is not perfect. So means the microbial process cannot produce the bulk chemical. Okay, bulk means microbial process can produce the small amount of the chemicals only compared to the chemical synthesis.
Oh, I see, I see.
Yeah. So typically the microbial process is applicable to the production of the, so you know, like how can I say, fine chemical. Okay, fine chemical. Yeah. That can be used for the pharmaceutical product, something like that. That is the idea.
Oh, I see, I see. For pharmaceutical applications, right?
Yeah, it is.
I see, I see. Very good. Thank you very much, sir. Thank you very much.
Yes, yeah, thank you so much for your great question.
Thank you.
Okay, thank you very much, Sensei.
Hi.
Now it's time for the question. You have the question?
Sorry?
Have the question from the
Yes.
audience, from the student?
Yeah. You they have. Yeah, if they have. Because the topic I covered this research talk is not familiar with maybe your student. But I believe the microbial process, using the microbes, how can I say, is so one of the techniques, how to produce the chemicals via the eco-friendly process. Okay. So if you have the question, please give me your question.
Yes, any question? So if not, I have the one simple question. So Sensei, what is the so thank you very much for your really nice presentation. What is the next or the future of your research?
Okay. Thank you so much.
It's been the next step.
Yeah. I'm also just focusing on how to produce the monomers, polymer feedstock. Then I just do it, just do it. But the other future plans, so I'm also creating the new types of the bio-based plastics, consisting of the resulting monomers, produced in my laboratory, like the itaconic acid or glyceric acid. They are very unique monomers having very great properties. They sometimes really hard to obtain as a reagent, meaning so we can't buy them. So I now preparing these kind of the monomers by ourselves. Then we are now trying to synthesize a new type of the polymers using these monomers we obtained. Then, so I'm also thinking of the collaboration with my colleagues, working with the polymer science and so characterization of the polymer properties, something like that. We are also going further to the next stage, regarding the polymer synthesis.
Okay, thank you very much, Sensei. What is about the polymer for beer?
Yeah, I believe. If so, it would be
Because
good.
you use many fermentation, right?
Yeah.
Okay.
So,
any question?
Yeah.
Any question from a student or teacher? So if not, on behalf of Faculty of Engineering and the course of Energy and Material Engineering, thank you. Many thanks for our professor also from KIT, Kyoto Institute of Technology, and also our close, more and moreover close friend, close friend forever. Thank you very much for every year that you give a lecture of this seminar. For the, especially for the student.
I appreciate again. You gave me this great time to share my current progress. And then I'm also looking for my collaborators. Working with the polymer synthesis using the monomers I obtained. And for evaluation of this the polymers to find out the new functions. Thank you so much. And then so I welcome you all to contact me.
Yes, thank you very much, Sensei. Hope to see you again.
Okay, see you again. Hopefully, so we will see you in January.
Yeah.
MSS.
Right.
And then have a safe trip to Hokkaido.
Oh, yes. Thank you very much, Sensei. Arigatou gozaimasu.
It's my pleasure. Okay.
どうもおおきに。
どうもおおきに。 プパササイプンゲンマ
คุณก็พูดภาษาไทยเก่งมาก
โอเค
โอเค
thank you
โอ
so
เค
much
บายบายเซนเซ่
บาย
บายบาย
บาย thank you so
Thank
much
you
ขอบ
very
คุณ
much. Thank you very
Thank you very
much.
much.
Have a good
so much
day.
thank
ขอบ
you
คุณ
so
นักศึกษา
much
ทุกท่านครับขอบคุณนักศึกษาทุกท่านครับขอบคุณและสวัสดีครับ
สวัสดีครับ
สวัสดีครับ